Try more. Hello, Nuber. How are you?
Fine,
And
thanks. And you? Hello,
yeah, I'm
everyone.
good.
Hi,
I'm good.
Saria. We are fine. Thank you. How are you?
I'm good too. And my lovely friend from Saudi Arabia, Ibtisam. Kaifa halik? How are you?
Hi, Sidi, how are you?
I'm good. I'm good. How are you? So, just waiting for the 3 minutes. Mhm. Just waiting for the 3 minutes to start, so I can take the feedback from everyone. If anybody would like to give your esteemed feedback up till now, how is your experience going with us? And of what were your objectives and any suggestions you want to add? Anyone?
I'm sorry, I missed what you said.
Yeah, I'm saying, can you please give us the feedback? How is the journey going of this summer camp? And any suggestions of yours that you want us to include in that summer camp?
I'm sorry, I couldn't participate very well. That's why I'm so sorry. I hope I can study and catch. I don't know. It's my, how can I say? I want to do. I want to learn so much, but I could not participate most of the lessons. I'm so sorry with this situation. I'm so sorry.
So, no issues, no issues. You can watch the recordings and get that in the WhatsApp group or directly to us. Or any suggestions? Any specific topic you want us to be covered in this summer camp? Tramor, Masibo, Rayaz Ahmed, Noor, Ajab Girls, and everyone, Akram.
I wonder if this course will be continuing or what's the last time, ending time for the course?
No, it will be continued. It will be continued until and
Okay.
unless we achieved all of the objectives of the course. That is why I'm asking the suggestions or any topic you want us to add in it, so we can add up in the summer camp. Today, this session is dedicated for the variant analysis, the mutational analysis, and regarding to that, we have a guest speaker talk, that is our invited guest speaker talk by tomorrow at the same time. So I would recommend everyone to participate in it, because that is actually the application part of the variant analysis, and that Professor Dr. Elias have sequenced a tribal genome, we can say that, a Pathan genome of his area, and he found some very interesting results out of it. So I would recommend everyone to please do join that session that we have arranged explicitly for you guys. So today we're gonna cover about the variant analysis, the SNP analysis, mutational analysis, and tomorrow we will be having the applied talk on that, the variant analysis, how you can uh, how you can study about that, and what are the related studies are being held according to the variant analysis, right? So, and that is that we will be covering the variant analysis this week, and then by the next week, most likely we're gonna go for the metagenomics, the bacterial one, and then we're gonna go for the gene expressions in the later weeks. Okay, so without any further ado, I can start up my session. So I would like everyone to have these files. Let me share my screen. You should be having the the chromosome 22 that I have shared before in the group. Like these two files, that is chr22.fastq and reference underscore chromosome 22. These two files everyone should have in our directory and name that directory as the variant. So can I expect that everybody do have that file? And also the variant calling single mDNA. I have shared in the group. So everybody do have these files because we also performed this variant
Yes, we do
analysis
have.
last week. So I'm gonna go cover some details today. Everybody do have that files? Everybody do have that files?
On my side, I have it because you
Okay.
sent us before, maybe the last week sent us CR22 and also a reference and also the words that contain about variants, polling single and database.
Exactly. That's what I want you guys to have. Okay. So that is good. That is good. So I just want these three documents: this chromosome 22.fastq and reference, and then. So today, just briefly going through what we're gonna do. Um, that is actually the pipeline of the whole exome sequencing or the whole genome sequencing. So you're gonna first and the four. Can anybody tell me about the? I'm just jumping into the NGS data analysis, and you are doing it for like a 1 month. We have started this course. So now my question is, can anybody tell me about what is the first step to do whenever you are going to start the NGS data analysis?
Quality control.
Very good. To do the quality control. And the tool for the quality control is, what is the
Fast
tool for the
QC.
quality control? Fast QC. That's very
We
perfect.
can use
Very
fast
perfect.
QC or multi QC both.
Yes, exactly. So we have multiple tools, for example, like multi-QC and the fast QC, as you have mentioned. But today we're gonna use the fast QC. So as I have mentioned earlier in yesterday's session, and before that too, that whenever you jump into the NGS data analysis, the first and the foremost step is to do the quality check. Either you get the data from the sequencer, is it of the good quality or the bad quality? Okay. So today's session is all about the pipeline regarding to the whole exome sequencing. So I'm gonna just open up my Ubuntu. I would like everyone to open up your Ubuntu and have those files in the desktop in the folder name as the variant. Okay, yeah. So I started my Ubuntu. I'm gonna set up my directory first, where my files are present. So for that, I'm gonna go for the cd slash mnt mount my device, and then I'm gonna go to the C. I need to check where my files are present. So right, just go to the files, right click on it, and go to the properties. So it shows that C users Sadia, OneDrive, desktop, and wait. So that is actually my whole address that I need to follow. Then C users OneDrive, no, Sadia, yeah, OneDrive, and then desktop, and then Variant. Perfect. Yes. And then yes. Uh, I have reached my directory. I can check where I'm exactly present. I can see from here MNT, see user Sadia, OneDrive, desktop, and Variant. But I can also check it through the PWD, enter, and it's also showing me my path, my folder, my present working directory. Right? So my directory is set. Now I just want to see which files are present. So I want to do the ls list. Okay. So these are the files. I do not need Linux command for now, but I just would need the variant calling single integer data. That is the word file. And this one, chromosome 22.fastq, and a reference underscore chromosome 22.tc. So we're gonna deal with these two files. My question is, can anybody tell me about which file from the highlighted ones, which file is should be the input file that needs to be aligned, or the file I need to process through the fastqc, and which one is the reference file? Can anybody tell me?
With fast queue extension. Files with the fast queue extension.
Yes, exactly. So the one having the fast queue extension is the one having the input, and the other one reference. It should be in the fast A format, and then that is the reference file. Reference. Okay, I can go over just go through the files just by do the. Let me take your. Let me take a quiz how I can check the first few lines of the, I just want to see this file. How I can check, how I can read the first few lines of that file. Can you tell me the command?
Yes, the file name head file name.
Perfect, perfect, perfect. You're doing great. So hit chromosome 22.fastq, and this is how your file looks like. Uh, ID and read sequence plus ID repeat and the quality control. Okay. And I'm gonna check the reference too. So head ref tab. See, that's actually my reference. It has a lot of ends just because my my reference is of the humans, and I have taken the reference of the chromosome 22. We have different chromosomes, so I have taken the reference from the chromosome 22 because our file contains the read from the chromosome 22, and everybody knows that we have the telomeric regions, so that is why we have a lot of ends here. So that is actually the reference file. Okay, I just want to make one point. How I can get these sequences or the references, I just want to go here and um, you need to go to the Ensemble Genome browser. Just go there. I just want to make a point. If you are having we are having the chromosome 22 file, and what suppose you're gonna have some different file when in the future when you're actually doing the NGS data analysis. So how are you gonna get the reference from it? So that database that is in Ensemble Genome browser contains multiple genomes in it, multiple genomes references in it. If just open up, I don't know if my internet is actually supporting it right now. We can come back over here and I can just tell you about that. Till then, we can just go through the fast QC tool. So the command would be fast QC and the file I want to give, that should be the FASTQs format because that is my input file, the file that I got from the sequencer, my raw data, that is the chromosome, right? And just press enter. So it has just started analyzing of chromosome 22.fast. This is actually the human sample, so it will take some time, a bit less file, but when you do it on your sample, it will actually take some more time because the actual data is of a larger one. Right. So finally, analysis complete for chromosome 22.fastq. Now I can do the ls and press enter. Yes. So I got this, that is chromosome 22 fastqc.html and chromosome 22 fastqc.zip. This one. These two files, these two files I got from the fastqc. I can just go directly into my directory and can see the files present over here. I will just open up this file, HTML. Perfect. Okay. So most of the options are green, are good, but some of them like per sequence quality is somehow falling over here, and see there are some bad quality reads falling over here. That is why our sequence quality is also being compromised. Per sequence quality, um, that is okay. Per sequence content, and it actually appears when when the sequence starts. So it actually gives not a good resource. So, but at the end, it is fine. Like how many A's, as many A's, as many B's. We can even see that as many G's and as many C's. Purpose and content. So there are no n content in our sample. Length distribution is actually normal, 75 and 77. That is actually the 676. Length of the sequences is 76. Sequence duplication level. We do have multiple duplicates, but it's okay. And overrepresented sequences here, you can get if any adapter is present or not. If you get that a red line or blue line or green line, that means there is some adapter sequence present, and then in the command you need to mention that this specific type of the adapter is present, and then you're gonna remove that. And then overrepresented sequences, there are no overrepresented sequences, and adapter content is also good, right? So actually we can go forward with this quality, but we have some uh, reads that are falling down, but we can go with it. We can go with it. It's okay. But just to make sure, just to move ahead with the trimming step, I can do a trimming of it. Okay. For now, I'm just using the default trimmer. I'm not adding so much options. Just I'm gonna use the tool for that is fast feed. So for that, you need to install how to install sudo apt install fast feed. Okay, enter. It is requiring my password. NTS123. Enter. So I already have that tool that is fast feed. That is why it did not take some time. Otherwise, you need to install that tool as similar as you have done the rest of the tools installations, right? So I will now just you do the trimming. Okay. What actually trimmers do? We got some trimming. We got some bad quality reads, and it will automatically detect that if you do not provide any options. I'm just going with the default one. If the read quality thresholds lies within the region, within the criteria of the file fee, it will clip it out, it will trim it out. Otherwise, it will not change anything. It will not gonna trim or clip anything, right? So in the fast fee, we actually gives the raw data file, we gives the input, and here we also generate some output to get a new fast queue file to that is trimmed one, right? So I can do ls, and I need to do the fast fee and the trimming on this file. So what I need to do is just call the fast fee. I'm not adding some options. If you wanna read this tool, just do fast fee dash h, and you're gonna see a lot of options. If you see, if you have in the adapter content, if you have the poly G tail present in it, you can just trim it off. If you have some adapter fastest sequence, you will even trim it off. This is how you can add multiple options based upon your um, fast queue report. So see, disable quality filtering. You will be disabling the quality filtering. It will unqualified quality fed, and then length required. If you want to trim the specific length, it is also requiring you the description of it. The reads shorter than length required will be discarded. So the specific length of the see we can see that we have same read length in everything that was a 76, but in sometimes when we are having the bad library separation, then we need to remove the very short reads. So in that case, you're gonna use that option, and so far there are multiple options you can use that. Okay, there is the thread. That is thread if you are actually using the samples of the large sample size. So you can speed up the command by using the dash thread option, or just by pressing the dash W and just add 10, 20, 30, 40, 60, and you can even cut by the quality just like this. Okay, so there are multiple options further than that, but for now I'm just going with the default one. with the default. Control L to clear it out. So just fast P. I need to give the input and need to generate an output that should be the trimmed one, right? So fast P and dash I. Dash I is the option for the input. Input. So my input file was the chromosome 22 dot fast Q and that now I'm just setting up the default options and dash O just to make my command very much simple and easier. Dash O is the output option, and I'm generating an output. What it's gonna do? Firstly, will read this file as an input. It will, according to its threshold, it will check which reads are falling into its threshold that needs to be trimmed, that needs to be clipped. It will do that and give me another file, whatever the name I'm gonna suggest. So I will suggest that chromosome 22 trim dot fast. Do not forget to mention the extension. If you want to get in the FASTQ format, mention the extension. If you want to get in the GZIP format, zip format, then you need to mention the extension also. Okay? So I'm going like this, enter. So now, see, it is actually detecting the adapters. We did not tell him to find adapters. It is doing it with the default of. So we're gonna wait for it. Yes. Duplication rate may be overestimated since it's a single end data. Okay. So, and it is also showing that the Q20 bases were of this one, and the Q30 bases were of this one. So this is how it generated some report. And now I can again check the fast QC. I can again apply the fast Q on my trim file. I can check if any difference present in it. So I can see that there must be another file that is this one. This is actually the trimmed file. Okay, you can even compare the file size. That was that was something 215 MB, and now it is 200 MB. So some of the things they have clipped, and now I have run again. So that is basically the pre-processing step. You need to do the quality, and then you need to trim the files, and then you need to again check the quality if the raw data now, if the clean data now is actually good to move forward for the analysis, or should I need to remove something more, right? So, this one, chromosome 22 underscore trim fast QC. Now open it up. See per base sequence quality, he have trimmed most of the reads that were falling into the red region. So now it shows that the per base sequence quality is good. I can now move forward, right? So with this, I have done with the pre-processing step. Now I'm gonna jump towards what actually I was aiming for, the objectives I had. I will just go to the control L. Are you guys following me till now? Any questions up till now? Okay, good, Atika. Any questions, concerns, queries? You can post in the chat box. Dr. Wasim is here. You can respond in the chat box.
आज कवर कर लेंगी आपने वेरिएंट फिल्टरिंग भी करवानी है आगे जाके
जी मैं कवर कर लूंगी
व्हाट्स टारगेट्स
to do the annotations and to check out which to do the filtering criteria, to set up the filtering
आई
criteria.
थिंक डिड नॉट वी कवर
or
एवरीथिंग बिफोर like alignments and I think वो obtained the file with the variants आप PCF से आगे शुरू करें ना
you have done with the GAT scan, we're gonna do with the freebase.
अच्छा चलो go ahead I think you would probably need another lecture
Let's
चलो
see.
ठीक है
I don't know.
tomorrow is Dr. Elias
Yeah, I know.
okay
Okay, so I just want to go for the alignments now. So after doing the quality, you will jump towards the alignment. You need to align the reference file. You need to align the sample file with the reference one, right? So for that, I'm gonna use the BWA tool. Just gonna make the indexes of the reference. So BWA index and which file of my reference file, reference chromosome 22.txt, and I want to make the indexes name as the chromosome.indexes. So for that, I need to give an option that is -p space chromosome 22 dot indexes indexes, right? It will make the indexes. So that is actually the whole pipeline for the whole exome sequencing. It will take a bit time and then it will be done. Once the indexes are made, I will just simply align the sample file with the indexes. Okay, now my indexes are made, and I will now simply align my sample file with the indexes. So for that, I'm gonna use the tool that is BWA again, but BWAMEM, that is the aligner. And yeah, you can even, if you have some larger sample size, so you can even put the threads option in it just to speed up the command. Just to make it simple, I will just go with the indices, chromosome 22 dot indices, and chromosome 22. And now, in this alignment, I want to be using the chromosome dot fast q but rather I'm gonna use the chromosome underscore trim dot fastq because that is the new file, that is the trim file I got, the clean file, right? Yes. And I just want to I just want to save my output in the SAM format. So that will be the chromosome 22 output dot SAM. So the SAM is basically it is now started my alignment. So now SAM is basically the file that is sequence alignment oblique mapping alignment file that is human readable. We have covered this alignment file formats and how to read the alignment file in the last week. So after getting that SAM file, I will just simply convert that SAM file into the BAM file. That should be that should be computer readable. It will just change the SAM format into the BAM format. Whenever you convert the SAM to BAM, you can see the visible difference between the file size. The SAM having the larger file size, but the BAM is having the smaller file size. So this is actually friendly to process into the computer, and also the BAM file is not readable by the humans, but it is um, readable by the computers. That is the computer language. You're gonna see that you can just do the head space chromosome underscore output dot sam, and then you can even do the head of the BAM file. You can see that you can do that. You can see how the SAM file looks like that you guys know, and how the BAM file looks like. It will some of the letters that are not readable by us, but it is actually known by the computers.
Hello.
Hello.
And if you don't have a reference uh, genome, then how we can find this output SAM file?
Um, that is actually the alignment file. So for the alignment, you have to have the reference file. If you do not, if
Either
you
you
do not
can
have the reference file, then you
let
can
me.
go for GG.
You can go for a
Let me.
closely related species. Okay. Okay. If you really want to do this variant calling thing, so whenever you do variant calling, you need to have a reference. If that species does not exist, either you go with assemblies and construct the reference, right? Okay. Like for
तो इससे
example, you
पहले
have
हमें
10,
assembly
10
बनाना है सर.
samples, 10 samples of the same organism. So you first construct a reference yourself, like you do the assemblies, and then use that. That will be the consensus of all those 10 or whatever samples you have. And then you can go align your individual samples on it and try to find their variants, right? So variants are always found when you compare your sample with some references. One approach is like you try to go with some closely related species, or maybe like if you're going for the human genomes and you're looking for your own genome, Pakistani genome, you don't want to compare with the reference all over the world, which is the consensus reference in suppose that has mainly initially it was the thinking that it has mainly Caucasians or Americans or Europeans. So different communities are developing their own references. So maybe you can go to close geographical location and pick up the reference like Chinese if it is available or Indian if it is available, and then you can compare your samples with that. Variant calling is comparatively easier as compared to genome assemblies, right? Especially with the eukaryotic genomes. That's really tough. Yeah, you have a comment? जी आपके मजीद कोई कमेंट था इसके ऊपर अजब who asked the question?
नहीं, मेरा यही कमेंट था कि अगर हमारे पास हमें पता ही नहीं होना कि ऑर्गेनिज्म के रेफरेंस जीनोम कहाँ पे हैं, तो हम डायरेक्टली असेंबली से जाके हम स्नेप और जो है ना इंडल्शन आइडेंटिफाई करना चाहते हैं, तो इसके लिए हमें सेम फाइल की
अगर तो
जरूरत
आपके
है।
पास एक ही सैंपल है तो फिर तो उससे कोई वो करना कोई इतना मायने नहीं रखेगा, लेकिन अगर आपके पास ज्यादा सैंपल्स हैं तो आप उनको मिला के उनका एक कंसेंसस डेवलप कर लें, असेंबली कर लें, लेकिन असेंबली इज नॉट अ गुड it is very tough thing to do, not especially with the eukaryotic genomes. So we die of doing that. So rather than that, we can take any closely related species or race whose genome is available. तो हम फिर उससे कर लेते हैं उसको अलाइन, ठीक है?
मेरे पास होल जीनोम सीक्वेंस करेक्ट है बैक्टीरिया का, तो वो हमें
बैक्टीरिया का, और उस बैक्टीरिया के बिल्कुल क्लोज जो बैक्टीरिया मौजूद हैं आप उनके साथ कंपैरिजन भी कर सकते हैं, और बट उसके अंदर आप कर सकते हैं you can you are interested to go for variants or you are interested to go for their gene finding or AMR genes. In that case आपकी जो बैक्टीरियल डेटाबेसेस जो है ना वो बहुत सारी मौजूद हैं, so it will compare with them, उनके वो कॉन्टेक्ट्स बनाएगा, असेंबलीज करेगा और फिर उसमें से वो जीन प्रेडिक्शन कर देगा आपको यूजिंग एनी जीन प्रेडिक्शन टूल. ओके
ओके, थैंक यू.
थैंक यू.
Okay. Yeah, thank you for your comments. Now I will gonna simply convert my SAM to BAM. For that I'm gonna use the tool that is SAM tools. SAM tools view. And that is the comment I have mentioned that my output should be in the BAM format. That is why I mentioned the B, and my input should be in the SAM format. So that is why I have mentioned the S over here. And I'm gonna simply mention the chromosome. The chromosome 2. The input file. That's the beauty of NGS. The output of one command will be the input of the other command. So chromosome to SAM, and then that will be the gonna have the output. Gonna name it as the chromosome output dot BAM. Right? So SAM tools view. It will simply convert my SAM to BAM file. Dash B is that the output will be in the BAM format, and S that shows the output should be in the SAM format. And similarly, we have the input in the SAM format and the output in the BAM format. Chromosome. It will not give us anything to print here, but it will just convert the SAM to BAM. And after that, I'm gonna sort it out and simply throw the variant calling. After you are done with the there is another step before the variant calling. You can go for the deduplication step. If you're actually working with the whole exome sequencing or the variant analysis, so you can before going for the variant calling, you can do the deduplication of proteins. So I'm not covering it here. That is actually the extra part, just covering the mainly commands that is actually present. So keep in mind when you are going for the whole exome genome, so you need to remove the duplicates. And after that, I will just sort it out and then the freebase. I hope everybody do have the freebase in your systems. If not, then you need to run the command simple, that is sudo apt install freebase. Sudo apt install freebase. I can send it in the group. And meanwhile, I'm waiting. And also, after after you are done with the variant analysis, if you get the variant calling file, you can remove the variants. You can even filter out the variants by using the BCF tool. There is a tool that is known as that called as the BCF tools. You can use that and you can remove the variants that are having the quality less than 30. Less than 30. It will remove the quality. That is actually you can need to for going for the variant, you need to have the high quality variants so that you need to rely on. You are going to finalize some of the genes. You are going to finalize some of the mutations. You are going to give your words regarding that. So that should be highly confident ones. So the variants, if having the low quality, it should be removed. Should be removed. So for that, you're gonna use the BCF. All Linux commands are different for every species. Yes, it is just because the tools can vary from species to species. For example, if you are using bacterial one, then obviously the pipeline would be different. Maybe you can use Velvet or Spades for the assembly, but when you are going for the gene identification or you are going for the phylogeny identification, then you're going to use multiple and you're going to go for the annotation. So that in that way, the tools will be different. You can use the Troca, you can use Kraken. So every tool have its own speciality that is actually offering to you. So yes, every species there are some specific tools that are designed for a specific species. But there are some other tools like the aligners, like the BWA, you can use it for the alignment for whenever you're going for the variant analysis. So now if I do the LSI, I do have the SAM file where it is. Yeah, the BAM and the SAM. Someone and the number. I can do the head. See, this is how your BAM file looks like. You cannot even read it up, but the computer can. But if I do the SAM, you can read it out. It simply convert it into its binaries, right? Control L. And now, and the next step was to do the sorting. So I just call the SAM tools, just SAM tools, sort my BAM file, and now I will just use the input file that was the output of my other file, and the output that is dash. Oh, or you can even go with the greater than sign. The greater than sign also shows that you are suggesting a name of the output file. The chromosome 22. I can make it simple. Sorted dot bam. Enter. I am skipping the threads input option intentionally just to make the command simple. But if you can see the document I have shared, so I have also mentioned the threads option that will actually makes your command even more faster, right? So I have done with the sorted.bam. See, and now I am left with the um, variant calling, and then its annotation, and then variant filtering, just how you can filter your variants. Okay, so for the variant calling, I need a tool that is a freebase, that is very handy tool, easy to install, just like sudo apt install freebase. Enter password again. It did not take some time because I have already installed that tool. So for the variant calling, you need to have the input file in the sorted.bam format, right? And also you need to have a reference, and then it will give you the VCF file. So for the freebase, I will just you can read your freebase tool by going with the help file. See, this is how your help file will looks like, and you can see that. You can go with the usage of it. Just study, add another any option you want to add in it. Then you can make your command. I'm going with a simple one with the freebase. So the option that I need for the variant calling, I have mentioned that I need to have a reference file and an input file that is the format of the sorted.bam, right? So these two things I need. So freebase dash f. I'm actually specifying the reference. So dash f is the option for the reference. Chromosome going to sorted.bam. Sorry, I need to give the reference first. So that was the reference underscore chromosome 22.txt space, and the input file that is chromosome sorted.bam, and within inside that will suggest you the output. I will just name it as chromosome 22.vcf. Perfect. Enter. It will create me a VCF file that will be the variant calling format. So the story will not end up here. It will need the to filter out the variants. You need to have the annotated one, like which variant is to be considered and which variant is to be removed, right? So what I was yeah. I will just annotate the VCF file, and in that I will integrate some multiple databases. Like I will need to check it from the SNP, SIFT, Polyfin, Fathom score. I need to check its prediction. It's score based, the prediction based. They will tell me that this allele or this variant is that much pathogenic or that much uh, normal or tolerated. If I do the ls, then I got this file chromosome 22.vcf. So I can do the head chromosome 22 dot, that is something the header. I can better go with the tail. See, this is how I got my VCF file. It shows that chromosome 22, position number this, the reference having the C and the alternate that was the T, and again it shows the quality, and yeah, there are also multiple things like it has calculated the allele frequency of some Asians or South Asians, something like that. But don't need to, and also it also shows the thing it is actually we need to focus on, that is type SNP, that is single nucleotide polymorphism. And see, it is type is equal to del, that is for the deletions. There's a mutation and that causes a deletion over here. So you can even check the mutation type over here. So this is how your VCF file looks like, right? Now I gonna go for um the annotation. Yes, for this, actually, when we are working on the Ubuntu, and there is an annotation that is I actually prefer to use the ANOVAR for the annotation for the variants, just because, but it as a, you need to have the license of it. You need to get the license of it. What actually annotations does, when you are working on the annotations on the Ubuntu on the Linux terminal, you need to download certain databases like the ClinVar, like the DB Snap, and the ClinSeg. It will actually it will actually check that specific variant into the multiple databases. It actually depends how many databases and which databases you are going to integrate. And I believe the genome browser is clicked down because I was just trying to oh, I will just go for the genome VEP. You can even I am actually not I'm not I'm intentionally not doing the annotation on the Linux just because I have to download multiple databases. So that will be quite huge for my system. So rather than downloading the multiple databases, I am just going directly towards the Ensemble, on that is the online tool, and I will just put my VCF file over there. But I will recommend, if you are when you are actually going towards the annotation, I would prefer that you need to do it on the Linux terminal, right? I don't know if if this can anybody able to open up the Ensemble VEP, or their site is down a bit. Are you guys able to open up the Ensemble? I guess their site is a bit down. I'm just waiting for that. Meanwhile, you can also check from the Ensemble. Okay, it is opening in the SRN. Open the open up your Ensemble variant effect predictor. Give me a second. I can just bring my charger. The one who is able to open up, can you please share up your screen? Because I don't know whether the site is down at my end, because this internet is working fine. If not, it is if it is not working fine, then I won't be able to attend the session. Dr. Wasim, do you have your laptop with you? If you can, it's working. Okay, if you can share your screen for a while, I can take the session from there.
Say it again.
Do you have your laptop? I just want you to open up the Ensemble browser because my end Ensemble is not working right now. I don't know. That is just not because of the internet.
No, sorry. Maybe you can ask the participants. You can see that where is Ajab.
Yes, I can share my
Yeah,
screen.
Ajab,
Yes.
sure.
anybody
Dr.
else who can
Sita,
do it?
How to share it? What symbol? That sharing option is locked. What?
Showing option is locked.
Yeah, make him presenter.
Okay, Ashraf, you can now share your screen.
I'm trying, but it is not working.
I try from my side.
Yeah, sure. Make a presenter. I have made you a presenter. Akram, I can make you a presenter.
Can you see my?
No, I cannot see your screen. So Google is working fine, but Ensemble is not working fine at mine. I don't know what's the reason. Yes, Akram, it is now working fine. I can see. Perfect, perfect, perfect. Okay, this is how your ensemble. Thank you so much, Akram. Thank you so much. This is how your ensemble variant effect predictor actually works, actually looks like. So here we have command line tool. If you're gonna go for the command line just for the Linux, but I'm not going for the command line for now, but I'm going for the web interface. So you need to click on that web interface. Akram, can you please go to the that web interface?
Yes, I can go to the web interface and press on web interface.
Yes, press on the web interface.
Okay.
And can you scroll it down?
Now the variant
Yeah,
effect
yeah, perfect, perfect,
open. Yes.
perfect, perfect, perfect. Yeah. See, from there you have there the default species already be human, and they are using the assembly of the chromosome. That is the version of the 37 GRCH 37. Now we are using the 38th version. That is why you need to work on the Linux, and if you are actually relying on the online tool, then you need to rely on the that specific reference they have already mentioned, right? Now you can input the file. You need to input the VCF file that you have generated from here. Just click on the choose files. Yeah, you need to click on the, I can click on the choose files and
Yes,
upload your VCF. Yeah,
I
yes,
click and I will go to desktop and I will
yes,
go
click
to the
upload your
variants.
VCF file. Yes, VCF file.
I can choose anyone.
No, you need to upload your VCF, the variant calling format, the one we got from the uh, after the freebase.
I this Chrome.
No, no, no.
No.
Okay, I can share you the file in the group. You can just upload that file. Okay.
Okay. Okay.
Yeah, I have shared that file. Just download it and upload.
This yes.
Yeah, yes, this one. Just upload it over here.
So
You download it? Mm-hmm. Perfect, perfect, perfect. Okay, now you need to see. We have selected the reference. We have selected the reference ones. We have input. We have gave the input part of the VCF file. Now, what we actually need transcript database to use. Would we don't need to use any transcript. We're gonna go for the additional configuration. Just click on the identifiers. It is actually showing additional identifiers for the gene, transcripts, and variants. Click on the identifiers.
the first one.
Yeah, first one. Okay. It is actually showing you what actually you want, you need. We need gene symbol. You want to have transcript version. Okay, it's good. Just scroll it down. Then you need to click the variants and frequency data. This is how um you can add multiple options in it. So click on the variants and frequency data. You want to check its protein, just click on the proteins. Its UniProt ID. If that protein is present in the UniProt, it will give you the UniProt ID. Okay, now, okay. Just wait. Okay, now, pull it a bit up. Yeah. See, it is actually showing you want to have the synonymous variants. No, the synonymous variant shows that if there is a mutation and it is actually not causing the change of the amino acid, then that mutation will not be affected, right? That mutation is known as the synonymous. So we don't need to click on the variant synonymous. It's perfect. And then frequency data for the co-located variants. Perfect. It is actually shows the 100 genome global minor allele frequencies. Or you can even click on the exomes one. Nomad exomes allele frequencies. Click on the nomad exomes allele frequencies.
Zooms.
Yeah, frequent in the yeah, this one. Okay. Now, nomad structural variant allele frequency, you that depends on you if you want to enable it, if you want to disable it. Perfect. Then include the flag variants. Just click on it. Include the flag variants. We are doing the annotation just by clicking on certain of the options. But if you are using the command line, you need to download all of the databases first, and then you need to make a command. Okay, click on the additional annotations. It will give you the additional transcript, protein, and regulatory annotations. See if anything we need to have. Exon, transcript annotation, no, perfect. Just scroll it down, go it as the default. Go it as a default. Scroll it down, go ahead. Go with the default one, this one. We need functional effect, yes. Enable your functional effect. If that variant is actually causing the changes in the protein or not, then you want to enable it, just enable it. Oh no, let's scroll it down. Yeah, the intact one, the functional effect, enable that.
I choose the
Mhm.
enabled. Yes.
Yeah. You can use the if functional, go with the PubMed ID.
Oh, in the same extra field include in the enable.
Yeah, yeah. Just click on the PubMed ID, PubMed.
Yes,
Okay,
I
double click
choose
on
it.
it. Perfect. And then just scroll it down. And you need to go for the gene ontology, which either that variant is doing something, or its ontology, how it is working, and their related diseases involved. Just click on the gene ontology.
I
I'm
click
just
here.
selecting it on. Yeah, exactly. Okay. Yes, move forward. Clinical significance. Enable the clinical significance.
Enabled.
Yeah, enable. Okay. You can even minimum percentage overlap. You can go with the 80%. 80%. Okay. You can use different options. I am using the most common that are being used and the predictions one now. Now we are up to the predictions. Here it is actually link that variant with the multiple databases and will give you the prediction and will give you the scores, and based on that prediction and scores we're gonna identify whether that variant is the pathogenic or the normal. So click on the predictions. Yes, what we actually need. We want C, that is SIFT, polyphen, DBSNP. Yes, for SIFT we want prediction and score. For polyphen we want prediction and score. That is perfect. And DV SFP, you can also enable it, but go with the default one for now. Clinical prediction, and just click on the clinical predictions. Just below. Okay, you can gene name with the gene name. Okay, and click on the clean prediction.
Click above box.
The last one, the clean prediction.
I don't understand about.
Okay, just go with the default. Just go with the default. And now the filtering options.
I press
Okay, there. Sorry, there. If you scroll it up, you will find out the CAD, that is CADD. If you will enable it, it will calculate the score, that measures the predicted deleterious the how much the variant is deleterious or not. That CAD. You can even enable that, right?
enable.
It
Yes.
will, yeah, the CAD means the combined annotation dependent depletions, and it is the VEP score that shows the how much. Go simple with the in the first one. The first one. Yes. Okay.
Yes.
Scroll it
Okay.
down. Yeah, perfect. Let's scroll it down
Yes.
and go with the filtering option. See, the run sign is still not green. Still, we have to specify uh, some of the filters. So you can just do the exclude common filters, and then advanced options.
Advanced filtering.
No, not this one. The last one.
Yeah. The
The
last
la
one. Okay, the last one.
okay.
Advanced.
If you have no, now click on the run.
Okay, I click run now.
Yes. See, This is how you have just done the annotation. Okay, you can stop sharing your screen and let me know when it is done. Till then, I can share my screen and tell you about something about the variant filtration.
Okay, I will stop now.
Thank you so much, Akram. So nice of you.
Welcome.
Stay blessed. Yeah, that is
Thank you.
actually running. Now I can share my screen. Let me know when you are done with the this. Okay. So yeah, there is some of the slides in which you need to use the you are actually setting up the filtering criteria. We we have analyzed our sample. We have done the annotation, and now we are something criteria which things we need to exclude and which things we do not need to exclude. So there are some extra parameters present in my filters, and as I that filters for the command that I've used in the Linux. But for the VEP, we have just used the SIF, we have used the polyfen, we have used that frame shift if it is present or not, and also this score based and the prediction based. This is how the score based and the prediction based scores looks like. So that is the filtering criteria for the tolerated ones. Uh, we will see, we will call all the variants having the frame shift, whether it is deletion, insertion, and substitutions. If it is present in the intronic region and the NCRNL region, it is in the UTRs or upstream or the downstream, I will just put it in the normal. I will not focus on that. My focus will be on the exonic on site, right? And now the databases will give you multiple scores, like SIFT will predict some score. If the SIFT is greater than or equal to 0.05, that means that variant is normal. So you need to make a consensus and specify that there are, you can even take that SIFT, polyfen, and polyfen in the mutational tester. There are multiple tools that can predict the variant. Or you can even go with I've gone with the score based and the prediction based. If my criteria was if the shift is less than 0.05 and polyphon is greater than 0.15 and GERP is greater than zero, sorry, is less than zero. Fathom is greater than equal to zero and LRT is greater than equal to 0.01. Also, the prediction score SIFT is equal to T, that is tolerated, polyphon B, that is benign. So then, if this criteria matches, then my variant would be tolerated. Otherwise, the other criteria was it for the deleterious one. So for the frame shift, if there's deletions, insertions, substitutions in exonic, intergenic, and splicing regions. Also, the SIF score is greater than, is less than 0.05. Polyphon is greater than 0.85. GERP is greater than 0. Fathom is less than 0. LRT 0.05. So these are the databases. These are the variant prediction tools are the scores that will tell you about if that variant is actually pathogenic or debilitating. And now, this if the SIF is also shows the prediction as D, polyfen shows the D, deleterious or the pathogenic. Similarly, the Fathom shows the D, LRT shows the deleterious. So if they're all present, that means my SNP would be the deleterious one. Deleterious ones. That is the you can even go with the shift polyfen, score based, prediction based, and the frame shift type, and then the GERP score or the mutational score. Mainly we're gonna use the shift and polyfen, score based and prediction based, and then if you are using the if you want to do some ancestrally like evolutionary studies, then you gonna use the GURP score, otherwise do not use that. And then there is another tool that is mutation analyzer, that is MP and MA, mutation assessor, and MP, mutational tester. So mutation tester basically it shows that if the protein changes or not. So if the protein changes, that will cause the deleterious one. If the protein does not change, so it will actually shows you the change of the protein. So this is how you gonna select the um you're gonna select the filtering criteria, right? Uh,
Assalamualaikum,
is there any
Rabia.
questions? Yes, please.
This filtering criteria is same for the web-based or Linux commands.
Yes, exactly. The web-based tools, that the web-based annotation only gives me the file, annotated file, that will be the SIFT or the polyfin scores. Now I want to set up my filtering criteria to find out, to filter out which variant is actually the pathogenic, which variant is actually the tolerated one. So I can just take the thing that, for example, if the SIFT is less than 0.05, then I can say that this file is the that the variant is actually the pathogen. So I hope I have answered your questions, Dr. Arya.
Yes, thank you. I got it. Thank you.
By the way, मैम आज आपकी आवाज चेंज है.
Yes, thank you. I got it. Thanks.
You got it? अच्छा आज आपकी आवाज चेंज लग रही है.
I'm actually too much tired today.
Seems like this. Okay. So now, yes, I have opened up
Hello, ma'am.
hello.
What is the command for terminal line of VEP? If you want to run on the terminal line, what will be the command?
You need to first download that VEP, and then you need to integrate that databases. For example, if you need to first download the DB snip for that. And if you go to the ensemble, there is the command line there, the command would be present over there. Um, Otherwise, you need to download the VEP in the terminal, and then you need to download certain databases, for example, the DBSNP, the CAD, and the different databases that whatever you want. And simple, the command would be uh, called just VEP, and you need to give the input file -i and the output file. You need to give the reference file. You need to mention that which reference version actually you are using, and then you need to plug in certain databases, or yes, you also need to mention the input file format, that is in the VCF file format, and the output file that you need to specify that, and the plugins that you need to add, for example, the DBSnip, the CAD, the CleanSig, and the rest.
Madam, the command I have provided in the chat box, is it right or not?
VEP, for example, that is .vcf. VCF भी आ गई हमारे पास, annotated बिल्कुल ये वो भी आ गया. कैशे जैसे जैसे ऑप्शन ठीक है, ऑफलाइन भी हो गया. असेंबली, इसके अंदर you need to add your reference file, reference भी आपको देनी होगी. और इसके अंदर you need to add plugins. Plugins भी आपको देने होंगे. For example, the databases you need to add in it. Yeah, the databases you need to add in it. So that is actually required. Otherwise, the annotation would be of no purpose.
Ma'am,
Two
can
things
you
are missing.
provide the correct command? Can you provide the correct command so we can run that?
Sure. I can provide it to you in the group because I need to search from my documents the exact command. So I will share you in the group. We are interested to annotate the VCF file from the command line using NOR. Yes. Okay. For that, I need to use my office PC because we have that Anwar over there. And for using the command line, I have told you that I need to have multiple databases to be downloaded. And I cannot download all of the databases here in my laptop, but I can download that databases into my workstation. So maybe I can take um, 20 minutes, maybe it will take just 20 minutes to generate a command over the generate and execute the command and put all the plugins in that command. So maybe we can do that, or I can make a video for it tomorrow, but I cannot do it right now because I do not have Anavara in my laptop, in my Linux, and I do not have any databases I need to integrate in my laptop, right? So actually, the databases required a lot of the resources. Okay.
Actually, this is the
Would you
question
like
I
to
have.
would you like to do on your lab computer, the big guy?
Can I ask a question, please?
Yes, yes,
Yes,
please.
please go
I
ahead.
want to do it on my workstation.
I would like to ask, what are the system requirements? We are dealing with a lot of new um, programs to install and programs to use on other websites. What are the system requirements for this?
Dr. Wasim, maybe you can answer? Or should I?
You go ahead first. Depends
Okay.
on your data size. Like depends on how many samples you have, right? So how we calculate that? Ideally, one sample from human genome. If you have the genome, right, the whole genome, and then obviously we look for the coverage. So what we have for the human genome analysis is system with 128 GB of RAM. You can process it very easily because normally if you have one human genome size is 3.2 gigabases, right? One base takes 1 byte on computer. So multiply it with minimum coverage requirement is 30x. So when you multiply 30, that means it is 96 GB. So to process a file of size 96 GB, you need to have the RAM, that is more important here. That should be more than that. So 128 is the best which we can have for 30x coverage. That's what we had initially in our mind while we were working on human genome assemblies. And you can process one genome at a time. But as you go along, you realize like coverage we need to increase. That's about the assembly projects. Okay, when you talk about the exome is not, uh, you know, it's not the whole genome. It's, I think, approximately 2% of the total genome. So 2% of the genome is your exome.
Okay.
So for a whole
Right.
exome project at that specific coverage, you can reduce that to whatever your requirement was for the full human genome at 30x coverage. You can reduce to 2% of the data. Sadia, how big were your files when you came up with the whole exome thing? What was the file size?
Um, 2 GB of one. Like I have the parent data and the forward was the 2 GB and the other one was
Yeah,
the 2
so
GB.
to process that 2 GB and for variant and assembly and alignment, I guess a system with maybe 16 or 32 GB of RAM will be enough for exome, right? 16 is good, 32 is
But again, it
excellent.
will require a lot of time, a lot of time.
What about the transcriptome?
By using,
That
by using,
time depends upon the RAM, but time also depends upon the processor. The speed at
Internet,
which it processes. Oh yeah, for the internet,
electricity.
here this ensemble thing, you need internet, right?
Yes.
So what was the time it took for your each sample to process on that big machine? Like, is there any way
Um,
you can avoid the dependence on the internet?
yes, by using the terminal, you are actually avoiding the dependence on the internet, but I cannot avoid the for the electricity supply. I need to have that.
Yeah, electric interruptions are there. But suppose if you have the continued electricity, you have a good internet, like normally in the houses, we have better internet because in the offices, the nets are shared, the universities. So I think my house
I was
internet is, I
able
think,
to
100
analyze, and
Mbps.
in university, I
Mhm.
was able to do the analysis of my one sample. It took like 2 days almost because the
Oh, really?
annotations and the alignment took
On
a
the big
lot,
machine,
lot.
on the big machine,
Yes,
on the big
yes,
machine,
yes, exactly.
2 GB
Is
sample
Alignment, yes,
for
the
alignment.
sir,
alignment,
sir, is there
Just
alignment
any
and annotations took a
Ajab,
lot
Ajab,
because I
Ajab.
have integrated multiple databases in that.
Just wait. Ajab has something to say. Ji beta.
sir, is there any free cloud system to run these commands in that Linux?
Good question. Good question. So cloud, we never did that actually, but there is one student. She was telling she paid like $100, 100 US dollars, and you like somebody use the clouds and then they return her the data within maybe 1 day or 2 days. So you don't need to buy them and you don't need to do that. But obviously, like if you have a dedicated system, maybe you can spend more time on that. I think 2 to 3 days are not hurting you much, right? If you have time, so maybe you can process them
Um,
with the 16
I
or
think
30
so.
GB of RAM. Yeah.
For VP, I think so. There is also, we can also run this online by using the plugins. Is there any option, if I'm not wrong, to run on terminal
yeah.
line and put some plugins? I think so.
Uh,
There is something, if I'm not wrong.
it should be something like where you can use the clouds and you can purchase them and then you can do that. So
No,
we
no, sir.
never did that.
सर
There
ये
is
जो
another
वी
cloud that
वी
is the
वी
DNA,
वीपी
DNA
को
Nexus.
क्या हम
DNA
टर्मिनल
Nexus.
लाइन पे
Uh,
By the way, tomorrow this guy is joining, Dr. Elias. So maybe we can also seek his guidance as well. He's doing exomes a lot at Omics Center. So
okay
probably he can have some clue on that. I think they have some local resources, but they might be using clouds, etc.
there are some of the clouds that are using for the NCS pipelines. They have the integrated pipelines over there, and one of them is the DNA Nexus. That is actually the commercial-based cloud platform, but it supports the full NGS pipelines, like for the alignment, variant calling, and the annotations. And yeah, for the variant annotation, they do have the VEP pre-installed in some of their workflows. But obviously, you
Mhm.
need to pay that. And there is another cloud computing that is the
Can they go
seven
on something like Galaxy or some online servers?
Galaxy
what
is
about
not the cloud one. No, 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 no,
uh,
no, no, no.
can can we run
Yeah.
the Linux command on Colab, Google Colab?
You can do that.
Yeah, so over there, I think you can purchase their. They give you the GPUs, right? GPUs are faster than CPUs, so I think you need to purchase, and then you can run the things on Colab. Yeah, you can embed the Linux codes in Python, and you can run them through Colab or Jupyter Notebook or anything.
सर वो
Yeah.
परचेज से ही भाग रहे हैं सर.
Yeah. Yeah. Chalo, wait karo. Dr. Silvano is coming in November, inshallah. So we will have its second variant. We will pick few of you in physical, but maybe everybody can join online one. Online is open, but physical we have only 25 to 30 spots. So we would encourage, especially the ones who had taken this course very well. They must apply for it, and I would recommend them for them to be selected for that physical one. So that guy is at ICB Italy. Maybe we can ask for his guidance as well. In the second part, we will go over the Anaconda, Python and Anaconda. So that provides you the environment where you can do multiple things together in multiple programming languages. Right. Okay.
वो जो आपको सैंपल साइज है ना उसको प्रोसेस करने के लिए वो तो आपको रैम तो रिक्वायर्ड है बाकी डिपेंडेंसीज के लिए यू कैन यूज द ऑल्टरनेटिव वे आप रैम ले लें या फिर आप क्लाउड कंप्यूटिंग खरीद लें तो वो you need to purchase that. You cannot run away from that.
Okay.
Yes. और एक और चीज ये है कि गूगल कोलैब के अंदर भी you need to purchase that. Yes. एक और ये है कि if you are directly connecting, you are actually working on some online data sets, then you can directly connect the collab with that online server that is there from where your data is present. Then it will not take that much of the RAM. But if you are using,
if we can upload our data to the Google Drive and then run from there,
that will take time. It should run that. It should run. Then you can use the Python and the to run your Linux command. It should run that. आप इसको पैरेलल प्रोसेसिंग की तरफ मत लेके जाइएगा ना आप एक एक को रन करते जाइएगा.
Okay.
So I think it should do that. तो जो उनका कुछ टाइम का फ्री सर्वर जो होता है ना उस टाइम के अंदर you can do that. You don't need to take some extra from there.
I will try.
Okay, perfect. Okay, I was just covering about the filtering of the variants. This is how I got my my file that was that I got from the VEP. So see, this is how you got the SIFT score, and this is how you got the Polyfin score. SIFT score and the Polyfin score. Now the T means the tolerated, D means the deleterious. You can apply the filters over there. Just go to the data. I'm just gonna apply filter. And I will just want to filter the T. Okay. See, this is how I'm filtering it out. And then the SIFT score was like, that is very much small file, no? So that is why I am actually doing, sorry, I have filtered out the tolerated one. I should not do that. This was something like, I should keep the D here, okay. So I got multiple Ds. Similarly, in the polyfen, I should go for the polyfen. I should go for the D or P both, right? तो वो D और P में दोनों ही आ रहे हैं मेरे पास D और P ही है परफेक्ट. अच्छा जी अब अभी देखिए मेरे पास जो फाइल साइज था ना वो कम हो गया मेरे पास जो नंबर ऑफ रोज थी वो कम होगी अब मेरे पास एक वही है जिनके अंदर पॉलिफिन पे मैंने फिल्टर लगा लिया सिफ्ट की प्रिडिक्शन पे लगाया और पॉलिफिन की प्रिडिक्शन पे लगाया अब आई एम यूजिंग द फिल्टर ऑन द सिफ्ट स्कोर I can go for the SIFT converted score, the best ones. So यहाँ पे I can, which one I need to do? SIFT. That was less than 0.05. So SIFT converted. So less than 0.05, I will select only the one which are less So in 0.05, rather, I can deselect everything behind it. Click OK. So this is how I can, I ended up with only three filters, or three genes over here. Obviously, my files include some of the variants that the file size was very much low. And see, this is how you got some of the exonic ones, the deleterious pathogenic. So this is how you can add the filters over here, and you got the filtered file. At the end, I am ending up with the three ones. So what these three filters are, what these three variants are doing, three genes are doing, I will just go for the go ontology. I will just take this, or maybe I can take some of the genes. Like I will, once we are done with the filtration, you need to copy the genes that you have filtered out, and then you need to paste it on the Go terms. I will do that. But for now, I'm just taking all of the genes because my file size is very much slow. I have already taken the 53. I can take it over here. I can get the unique ones is equal to unique. Just teaching you how to perform the Go term analysis. Control C, and I can do it like paste 1 to 3, or इसको Control C कर लेते हैं. Now I'm gonna go for Go term analysis just to find out which pathways, which ontologies, which diseases are present. For that, I'm gonna go for the enricher. Enricher. This one. I can share you the links over here. Yes. So it is just asking me to input the data. I will just paste my genes over there. Genes, copy, and paste over here and submit. Obviously, it is these genes are very much slow, and I will just go to the ontologies. The number is very much low, so I need to maybe it will not give that much or the maximum of the things, but I will just go for the Go biological process, and it shows that maximum of the things are involved in the blood coagulation, intrinsic pathway, protein activation cascade, and that is the bar graph. I will just want to know what genes are actually present in the pathway. I will just go to the clustergram. It is making up the clusters. So see, so these are the FLNA that is involved in the blood coagulation intrinsic pathway. FLNA is also involved protein activation cascade. FLNA also involved. So the green line that is actually making a heat map. Wherever the red mark is present, that gene is present. That means the MLHE gene is present in the positive regulation of oxidoreductase activity, and NOC2L, it is present in the negative regulation of lymphocyte. This is how you can check it out. And then obviously you filter out your genes that is relevant with your disease. And then you can correlate with the literature. You can make up a story. This is how you can do that, right? So by here, I can end up my session. So this is all about the variant analysis, variant filtration. Now I am very much open for the queries, concerns, and stuff.
Excellent, Sadia. Wonderful. This was your soul of light. Yeah. And thank you so much, the participants. Your questions, your concerns, and comments. They were really interesting, right? Some of you had been enjoying this thing very much. I can see you.
Exactly. And Really, their questions are very much insightful. बहुत interesting से questions होते हैं. I hope everybody enjoyed today's sessions. You can ask the queries, concerns. Yes, I am 3 minutes ahead of time, so I have
मैम अगर
input.
ये वीईपी का जो वीईपी का कमांड लाइन की जो
वो मैं आपके पास share
कमांड्स
करती
है
हूँ.
वो अगर सेंड कर दे तो फिर हम कोशिश करते हैं रन करने की.
मैं आपके साथ वो भी share कर दूंगी. मैं आपके साथ I can
Which
share you some more ones.
commands? Which
VEP.
commands?
वीएपी नोटेशन कॉलिंग.
Oh, yeah, yeah. We'll also cover in the RNA-seq as well. So what you do is you take the genes and you try to correlate them with their functions, right? So which functions these variants are related to genes, of course, right? And those genes are related to which functions. Then we go into those variants and we see how they are changing the proteins. Obviously, one variant might change the amino acid, and that amino acid, if it is critical, can change the protein structure, right? So that's why they are damaging. So which of the functions have been interrupted by these variants? That's important. So this thing is called as Go term analysis, gene ontology terms, right? Go terms, GO.
अस्सलाम
Okay.
वालेकुम सर।
Walaikum assalam.
सर कैसे हैं आप? ठीक हैं?
जी बोले.
अच्छा
Fine.
सर, मेरा सवाल जो है वो ये है कि VCF फाइल जो है इसमें अगर इन केस हमारे पास इन हाउस डेटाबेस है और हमारे पास 1000 में एग्जॉम है और हम अपने VCF फाइल के अंदर अपना इन हाउस डेटाबेस जो है उनके वेरिएंट्स हम अगर इंसर्ट करना चाहते हैं तो उसके लिए क्या तरीका होगा? और दूसरा सर ये कि जो VCF फाइल आप लोगों ने जनरेट किया हुआ है इसके अंदर होमोजाइगस हेटेरोजाइगस मतलब इस तरह का कोई कंडीशन वो भी मैंने नहीं देखा इस फाइल के अंदर।
Yeah, we did not go. His question is like, what about the heterozygosity or other filters? So we for the analysis and for the thesis and the paper, we obviously go with that. We have to look for, I think, some percentages of this, allele frequencies. If they are more than, what was that, Sadia, 5% or 0.05 is 5%, obviously.
Yeah,
So then they are
5%.
not variants. We can call them as polymorphism. So we apply all those filters. And by the way, you applied the filters not using Excel, right? In your real work, you applied them using, I guess, R scripts or Python scripts. So while
Yes.
you do this filtering in Excel, and somebody asked a really good question. So normally you have like 150,000 variants in a big file. So if you open up that Excel file, obviously in Excel you have to open the file and then you need to work on that. So while the file is opening, it's slowing down your system, right? And sometimes your system even hangs. So why don't you come up or you write some scripts? Normally you can do that in Linux
Sure.
using shell scripts, but he also has Python and R code. So when we cover R and Python, we will give you those scripts as well. Okay. Now coming back to your own local database, which you want to use for this variant filtering, obviously you can do that. You can do that, but you need to then write your own scripts to incorporate your own DB into that,
Yes.
right? So right now we are using the online things which are already available and their databases which they have on the back end. But if you have your in-house database, you can obviously use that because here you are filtering your variants against the variants coming from this population, this local Pakistani or local regional population, but that only resides on your database. If you share your variants with them, so over there they also give you an option to, go to area specific databases. But unfortunately, our people they don't share their data with them, so it's really hard to, you know, compare them because as people keep on reporting novel variants from Pakistan, they keep on adding into the databases. So when some variant is already reported and when it is present in a significant amount, then it's no more a novel variant. It becomes a polymorphism in that particular population. But obviously, our data sets are not there yet. So we are supposed to compare which DB offers from all across the world, right? So you need to come up with your own pipelines or your own scripts to do that. So that will be a little bit hard for now, or a long way to go.
Uh, yes, thank you so much. And just adding a comment to that, the heterozygous zygosities one, for that, we actually use a database that is the NOMAD. We usually write it as the GNOMAD, genome aggregation database. So that will tell you about the heterozygosity and the homozygosity. Another thing, DB step can vary the exact that are actually we're gonna use, but for this specifically for the zygosities, we use the NOMAD GM. I have mentioned in the text box. That will tell you about the whether the allele is the homozygous or the heterozygous. And the Hamdia is also saying if
Okay,
you
so
want to learn, well, okay, I will just I can share you the codes. Maybe maybe I can find it out right now. So yeah, I can share you my screen. This is how I
after
use
this
the
session, or maybe in parallel, you guys can make different groups. People working on exomes, they can get together and they can continue on that thing. Share with each other, share with the community, right? And help each other. Similarly, the metagenomics, I think that's our next week topic. I'm delaying RNA-seq. These girls are really pushing me back because, and then I have to do start the other course. But I think one of the participants, she was very keenly interested in doing the metagenomic analysis. So we are bringing in next week metagenomics, and then RNA-seq goes, I guess, one more week ahead, and then we will finish, inshallah, this course. But obviously, I see that as long as you people are enjoying and you are loving it, we are in no hurry, right? But this is just the beginning. For some of you, some of the advanced users are also there, so they can help the others, right? And this way you can develop a great community with each other.
yeah. So I have printed
By the
out
way,
the PDF.
this gentleman who asked
I
the
think
question
so.
about this
I
DB
think
thing,
so.
you
Akram
can
have
also
completed the task. So
very
I think
wonderful.
so.
Akram.
Akram have completed the task. Can we see his screen?
Yeah, just 1 minute. I need to make a point for the Hamdia. She's interested to how to filter out the variants using the Python. So that is the code I have used and I'm using for it to to filter out the variants. You need to just specify the filtering criteria, whether it is of the frame shift. That is totally depending upon this table I have made, and then this code. For the first filtering criteria, based upon their function.ref gene and exon function, I basically put the header in that, which columns to filter out, and what exactly there should be present to, and then which thing I need to get and which thing I need to remove. And that was the first filtering criteria. Then the second was on the based on the scores, that SIFT score hai, itna hai, polyphen itna hai, then you need to mark as normal. If there is SIFT score is less than 0.05 and polyphen yehi hai, then you need to return it as pathogenic. And then the third labeling criteria was on their prediction based. Similar on this Polyfan Fathom mutation tester and mutation assessor. You need to return the pathogenic and the normal.
You
So
can
that
cover that when we go to Python. So she just need to wait.
okay, sure.
Are you
Okay,
working
sure.
on Python, Hamda? Who's there? If she's not familiar with Python, you can
Yes,
hold. Yeah,
she's
yeah.
in the textbook. So Vakarian ke, yes, she's familiar with the Python and things too.
Oh,
Sorry.
perfect, perfect. So share with her so she can do that. Any problems? Get hold of Sadia.
Thank you so much, Atika. Um, yeah. Starting from fast to VC, thank you. Yeah. So now you can make a full pipeline. That will be easy for you to cover it up. Thank you. Okay. It's it's like 8:40 now. Any queries, concerns, you can share in the WhatsApp group. And
Good night.
um, good night. Yeah, I little need a good break now. Thank you so much, everyone. Just post your queries, concerns in the group, and see you inshallah tomorrow with Dr. Elias sessions. Thank you so much.